In this presentation, I'm going to talk about the pangenome analysis of angiosperm plastomes using PGR-TK. Let me start with explaining some key terms. Pangenome refers to the comparison of a large number of genomes. Angiosperms are flowering plants, plants you are all familiar with. Plastomes in this context refer to chloroplast genomes, since all green flowering plants have chloroplast in their leaves. Those chloroplasts are responsible for photosynthesis, producing food that both the plant and we benefit from. Finally, PGR-TK is a software tool that I'll discuss in more detail shortly. Now, why are we doing this and how did I get started on this problem? As many of you know, a lot of researchers are currently analyzing genomes to better understand their evolution. A common approach in these studies is pangenome analysis, which has largely been applied to humans and to a lesser extent to plant and other organisms. And that brings us to the important question, do we need to focus only on the human genome to understand the human genome? Of course, human genome is where the money is, and you can see an interesting parallel from an earlier era, from the medieval era, when alchemists convinced kings to fund their work on turning other materials into gold. It was truly doing research where the money was, because gold was money, and so the kings invested in those efforts. However, real scientific progress in understanding materials came much later, when physicists studied the hydrogen atom, not because hydrogen was valuable, but because it was the simplest case that helped explain more complex systems. Following similar logic, I began with bacterial genomes. Bacterial genomes offer a better starting point for understanding genome evolution, not only because they are smaller, but also because we have access to many diverse sequences. Since all organisms' genomes follow the same common principles, I believe that starting from bacteria would allow us to scale our understanding up to unicellular eukaryotes and eventually to humans. I spent quite a bit of time developing tools to study bacterial genomes, but soon realized that I needed a good visualization tool. That's when I reached out to Dr. Jason Chin, who was working on human pangenome analysis and developing a visualization tool called PGR-TK. With his help, I learned how PGR-TK worked. And then during the summer of 2023, I worked with a number of high school students using PGR-TK to analyze chloroplast genomes from 5 different genera of plants. Since then, we scaled up the analysis to understand the plastomes of all flowering plants using PGR-TK. That work now reached a mature stage, or what the computer programmers call release 1.0. In this presentation, I'll talk about what we observed. Why chloroplasts? There are several reasons. First, it's worth noting that they are actually derived from bacteria. Chloroplasts are cyanobacteria which got engulfed by eukaryotic cells billions of years back. These cyanobacteria could perform photosynthesis, converting light into food. Over time, they established a symbiotic relationship with the host cells, leading to what we now recognize as chloroplasts. This endosymbiotic event happened in an ancestor of modern plants, specifically green algae like Chlamydomonas, which you can still find in the oceans today. Also, these chloroplasts have their own chromosomes, which are different from nuclear chromosomes. Nuclear chromosomes of eukaryotic cells are linear, whereas chloroplast genomes are circular like bacterial genomes. In terms of sequence size, these chloroplast genomes are around 140,000 to 160,000 nucleotides long, which is one order of magnitude smaller than the bacterial genomes. So chloroplasts offered a sweet spot large enough to find meaningful patterns, yet small enough to be manageable. Chloroplast genomes are widely available from a diverse range of plants, which themselves survive in an array of environmental conditions. This diversity makes plastomes a great subject for studying genome evolution. Typically, plant plastomes are around 140 to 160,000 nucleotides long, like I mentioned before. However, there are some exceptions. Some plant plastomes extend up to 200 to 240,000 nucleotides. I don't show them here. While others, particularly these parasitic plants, have drastically reduced plastomes as small as 30 to 40,000 nucleotides. These parasitic plants rely on other plants for survival, so they lose part of their plastomes over time. Otherwise, plastoms are generally highly conserved, which make them particularly interesting for comparative analysis. So we hope that by studying these, we would not only find patterns but also gain insights that could inform future work with bacterial genomes, and which is a topic we'll be starting this summer. So interestingly, plastoms turned out to be more complex and fascinating than I initially thought. So we continued to work on them for one more year than our initial plan. Now, as for our methods, we downloaded all available chloroplast sequences from NCBI. Then we filtered the data and retained only those from flowering plants. We organized the sequences by genus and family and only kept groups with 3 or more sequences. Then using PGR-TK, we ran pangenome analysis on those subsets. Now in the next part of the presentation, I'll walk you through some of the common patterns we observed in these plastoms and what they might reveal about the genome evolution of plastids. Before I show you the actual images, let me cover one more topic that will help you in understanding the PGR-TK plots. Here is the structure of a typical angiosperm chloroplast. You will see that there are 4 blocks. There are these 2 inverted repeats. Inverted means they are in opposite strands, and in between these inverted repeat there is a small single copy region, and on the other side of the inverted repeat is a long single copy region. This is the conserved structure that you will find in all chloroplasts of angiosperms. Research shows that it came from cyanobacteria that got engulfed into the plant cells. In the next few slides, I'll walk you through a few typical PGR-TK plots. Let's start with Vitis. This is the genus that includes grapes. Here is the PGR-TK plot. Since this is the first one you are seeing, let me explain what you are seeing here. On each line, this plot shows a plastid sequence. So every single line is a plastid sequence, and here it mentions which Vitis species that is being shown here. So the first one is Vitis Shuttleworthy, and then it goes like that. Now, you have to also understand that each of these line or a plastid sequence is a circular sequence. So the plot shows it as a line, but the sequence of this green part is followed by the yellow part. So if you also remember the typical structure of a plastid sequence, it has long single copy region, short single copy region, and inverted repeats. On PGR-TK, the software allows me to click on each segment. So I click here. Now, these different lines represent different plastids, and what PGR-TK does is to find the conserved sequences among all these plots and then show them in different color, which means all these highlighted sequences I just highlighted now are conserved or same sequences. And here you see that I highlighted this portion and it also picked this because this is the inverted repeat. So they have the same sequence. And you know that it's inverted repeat because if you look at the arrow here, it points to the left and this arrow points to the right. The specific direction doesn't mean anything, but when you have arrows pointing in 2 different directions, that means these sequences are reverse complement of each other, as it happens with the inverted repeat. So this long region in between the inverted repeat is the long single copy region, and this blue, green, and then it comes with this yellow, that's the short single copy region. So you can see that it's quite uniform. All of these sequences have very similar structure. If you go down, there is one sequence that appears to be unusually long, but I think that is an error, some kind of sequencing error or something happened with this particular sequence. So that should be removed. I didn't do it just to show you that the kind of errors we often get. But usually you see that most of these other sequences are of same length, and that length is usually around 150 to 160,000. That's a typical length of plastid. Sometimes it can go to 110, 120, but it stays very close within our range. This one particular sequence that got too large gives you this range of 240,000. So on this top, you can see the scale. All right, on the left side, PGR-TK also generates phylogeny based on those sequences. So it PGR-TK's own algorithm through which it generates the phylogeny. Let me move on to another plot. I'm going to next show you magnolia, which is a flower that you are familiar with. So here is the PGR-TK plot. Once again, you see that here you have, if I click here, then it also picks this region. So this is the inverted repeat. In between, you have the short single copy region, and here you have the long single copy region. Now, one thing I should mention is when I run PGR-TK on the original sequences that I download from NCBI, they don't align so nicely because each sequence starts from a different position of the circle that the plastid is. So what I've done is I ran PGR-TK to find these conserved places, and then I rotate the sequences so that they all start from the same PGR-TK block. Which block that is picked randomly. There is no fixed location in the context of these LSC, SSC, or anything, but they all start from the same PGR-TK aligned block, and then I rerun the software again. So that's how you get these nicely aligned plots. Okay, once again, with magnolia, you see that they are well conserved, except for one or two sequences. And once again, I believe at least this unusually long sequence is an error, but this one maybe has some significance. I'm not sure. Sometimes what I've seen is they are maybe not magnolia, but closely related genus, but got included in the plot because of misidentification. I'll come to that topic in a later part of the presentation. So on the right side, you see the names of these different species. They're all magnolia of various species name. And then the left side shows you the phylogeny. Let me move on to a third plot. This time I'm going to show you rubus, which includes the blackberries and raspberries and a number of other berries like salmonberry. So here is the PGR-TK plot. This one is a bit more complex in terms of fragmentation. That means the sequence is less conserved, but you can clearly see the inverted repeat. So here, if I click on this blue, it also picks this blue here. Also, if I click on this green, it highlights the green here. If I click on this, wait, if I click on this yellow, then it highlights the yellow on this side. So those three constitute the inverted repeat, and then this portion is the there is another portion of the inverted repeat, this one. So in between you have the short single copy region, and here this longer area. These sequences are the long single copy region. One reason the sequences look fragmented could be that there are more sequences, but that's not always true. You can see that in Magnolia, I have a similar number of sequences, but they are not as fragmented. What the fragmentation means is that the sequences diverge from each other. So if these various PGR-TK plots have less fragmentation, that means the sequences are more similar. So here it seems like these robust sequences are more divergent than magnolia or vitis that you have seen before. Another pattern you can see is that the single copy regions are more fragmented than the inverted repeat. Here you can see that the inverted repeat is only split into four rectangles or four arrows, whereas the short single copy region is highly fragmented. So that means the inverted repeat is more conserved than the single copy regions. And that's the well-known pattern. People know that in the field. So we see that confirmed by the PGR-TK plot. Now I'll move on to the fourth one. Next, I'll show you the genus Rosa, which includes all roses. Here is the PGR-TK plot, and let's start with the top. You can see that it follows the same pattern with all these names of the roses on the side, and here you have the inverted repeat. I'll highlight here, and you'll see that it also picks the region on the other side. This portion also is part of the inverted repeat, but in between you have the short single copy region, and here you have the long single copy region. But here in this Rosa plot, we see something different. If you go down to the bottom, you can see that the plot for these 15 or so plants are different from the rest. If I click here though, I see that this one is identical to here. If I click further, this one matches here. If I click here, it picks up all these yellow here. So essentially these 15 have the long single copy region inverted from the rest. What is happening here is that it's well known that in plastids, these sequences, the plastid sequences, are present in all plants in two isoforms. So in one isoform, the long or short single copy region is inverted with respect to the rest, and they remain in about 50-50 mixture. So it just happens that when the plastid sequences are assembled, the sequence that gets submitted picks only one of the two isoforms. And I think what happens often is that these sequences try to match each other from different species to generate the assembly, and that's why you see a degree of uniformity, like where you uh, check the rubus or magnolia, you didn't see the mixture of 50-50 of these two isoforms. So probably they had both of these sequences and picked only one of those two orientation based on uniformity among others. I'm not sure how it's done, but what I see from time to time is in some genera, a portion of the sequences have inverted single copy region, either the long or the short one, compared to the rest. They're not usually 50-50, they're usually like, say, 20-80 or 10% versus 90%, but I see some of them with the opposite orientation. And what I typically do is write another script to invert these ones so that they all have the same orientation. In the last four plots, you saw PGR-TK patterns for Vitis, Magnolia, Rosa, and Rubus, and they show the typical pattern. And my students and I scanned through all of these various plots manually. First, my students looked into 4 orders, and then I checked all of the rest. And what I found was about 75 to 77% of all genera that we looked into have those standard patterns where you can clearly see the inverted repeats, the short single copy region and the long single copy region. And then a number of the other ones do not follow that pattern. So let me start with geranium. This is the beautiful flower you are familiar with. Here is the PGR-TK plot. So once again, each line shows you a plastid. You can see that they're all geraniums. And here you see that it's completely messed up. First of all, they're really fragmented. All of these lines are fragmented, and only a few have conserved sequences where I can pick up these red lines in both of them or purple in both of them. And then here you have these green ones. But other than that, there are no conserved sequence among all of these. Maybe a small portion, but mostly they show highly fragmented and quite dissimilar patterns. Another one is this Zyris. It's a grass flower. Here is the PGR-TK plot, and you can see that it's completely fragmented. and you don't see the inverted repeats. We cannot find the inverted repeats from these plots. So what does higher level of fragmentation means? That means the sequences are quite divergent from each other. Here is the third one. It's Hydrocharis. It's an aquatic plant, and it's the genus or order Alismatalis. You can see the PGR-TK plot. Once again, it's highly fragmented. There are some bits of conservation like these blue, and then maybe these ones. And this is probably the inverted repeat because you pick this side, you get the other side. But other than that, here is some inverted repeat. But apart from that, the sequences are really fragmented. So this is a pattern I have seen in a small number of sequences, maybe 100 or so among all of these um, plots I manually went through. And after going through them manually, I also wrote some scripts to analyze or identify these patterns. The next pattern I will show you is quite puzzling, or it was when I first came across. This is the plant called cyclamen. Here is the PGR-TK plot. You can see that four or five of them match each other quite well, but one of the sequences is completely different. So here these four, these five on the top are similar. You can see that all of them have same kind of PGR-TK blocks, but the bottom one is completely different. If I click any of these blocks, nothing else gets highlighted, which means this sequence is very divergent from the rest. And at first, I had no idea what was going on, but then I took a portion of the nucleotide sequence from this one that is not matching the rest and searched at the NCBI. And what I found was it was matching a completely different genus apart from the sequence that it originally was. So when I did blast off a small portion of this sequence at NCBI, it matched the cyclamen persicum because that sequence is there in NCBI, this particular sequence. But the rest of the matches were on a completely different genus called Pseudostellaria. So what I did next is I ran PGR-TK by combining cyclamen and Pseudostellaria, and here is the result. You can see that the top portion has a set of patterns and the bottom one has a set of patterns. The top one is all pseudostellaria, the bottom one is all cyclamen, and this cyclamen persicum that wasn't matching the rest of the cyclamens now fits perfectly within pseudostellaria. And this leaves you with no doubt that this particular plant was actually pseudostellaria misidentified as cyclamen in the NCBI database. So this turned out to be the explanation for a large number of those completely mismatching plants. I made a list of 35 or 36 cases where one or in one case two sequence did not match the rest. The rest were all very similar. One sequence stood out. And in each case, I did this whole analysis. I took a portion of the sequence, searched at NCBI, and then whatever the NCBI result gave, I ran BLAST or PGR-TK with both sets combined. And in a large number of cases, I'd say 80% of the cases, it turned out to be incorrect annotation for the plant. The next group I'll show you consists of parasitic plants. This is a plant called Hydnora, the one that I'll show first. This is an interesting plant. It pollinates through making a smell that's attractive to the insects, and then it traps the insects and eats them. The interesting point for our purpose is that these parasitic plants have highly reduced form of plastids. So you can see here, these are the Hydnora plastids, the PGR-TK plot. the length is 30,000 nucleotides, whereas typically plastids in almost all flowering plants have lengths of around 140 to 160,000 nucleotides. So this is highly reduced. And when I draw the PGR-TK plot, I see that they don't match each other at all. So quite a bit of divergence can be seen. Now there is another one. This is an orchid called Euania, and it's once again a parasitic plant. This case, the PGR-TK, the sequences are about 50,000 nucleotides, so quite small. Here you can see that all these plants have similar patterns, and you can click and identify the inverted repeats. And this is probably the short single copy region, but this is definitely inverted repeat. So you can see that it has clearly identifiable inverted repeat, and then this portion is the SSC, and this longer region is LSC. So a little better than Hydnora, but you still see a lot of fragmentation. And the third one I'll show is Gastrodia. This is a potato orchid. This is another parasitic plant. And you can see the PGR-TK pattern. Here, three or so are quite divergent from the rest. The length of this is 40,000. So the top ones have a length of 30,000. And you can see that the top ones are also quite divergent among each other. Maybe a few are similar to the others. These two are, for example, pairs, but most of them are quite divergent. So when I come to these parasitic plants, I see a lot more divergence, but to understand what is going on will take a bit of analysis. So I haven't done that yet. At least I've seen the difference in PGR-TK plot. And finally, a small group of plots have all kinds of complexity mixed. Every kind of odd pattern you see in the previous one is present. So I'm going to show vaccinium, which includes blueberries and cranberries and a number of other berries. You can see these are the PGR-TK plots. First thing I notice is that they have quite divergent lengths. The lengths do not match each other, and the length is also higher than typical plastids, so it's almost reaching 200,000 nucleotides. Typically, it's 140, 150,000. And then, although there are similarities between these various plots, for example, you can highlight these sections, all these sections, but still you see that there are portions which are quite divergent. Like, for example, this particular one has all these sequences which look highly fragmented and divergent from each other. So I'm not sure what that means. To understand that, we have to go and look into the sequence at the gene level and try to analyze what is happening. The second one I'll show here, it's this is the rhododendron, and it's closely related to vaccinium in the same order. And you can see the top few ones have a lot of similarities, but the rest are all quite different. And the length, again, reaches close to 240,000 nucleotides, this long one, but the remaining ones are also probably above 200,000 nucleotides. I don't know the scale here, but I think it's close to 200,000. So a few of the sequences match, then there are all these short sequences and long sequences. You see these strange fragmented, highly fragmented regions. So it's not clear what that means, but at least through this PGR-TK analysis, we can narrow down the set with unusual patterns and then analyze them further using all kinds of other methods. All right, here I'm going to summarize what we have learned so far. We use PGR-TK to analyze chloroplast genomes from all flowering plants. We downloaded the sequences from NCBI and split them into different genera, and we got 944 angiosperm genres. And overall, we noticed 5 broad patterns. In about 75 to 77% of the sequences, we saw clearly delineated plastic structures with small single copy, large single copy, and inverted repeats. In a small number of the sequences, we found highly fragmented patterns, and then we noticed annotation errors or plant identification errors in a subset of sequences. And for parasitic plants where the genomes were highly reduced, we saw all kinds of mixed patterns. And finally, a small number of sequences had complex patterns, and some examples were blackberry and the rhododendrons. Those need to be analyzed further to learn what is going on. I'm not going to include the analysis of the plant families. We did similar analysis for the families just like the genres. I'll present that in a later video.